because the degeneration process do not stop, and other processes also involving other autosomes can also create multiple sex chromosomes by rearrangement between the sex chromosomes and autosomes. There is another way that sex chromosomes also different, multiple sex chromosomes also arose by fission of the ancestral sex chromosomes, and both these scenarios of sex chromosome evolution can be observed in Hartia. Hartia is a genus of armored catfishes widely distributed in South American rivers, and this genus is considered to be a monophyletic genus composed by three main clades with highly congruence on their geographical distribution and distribution of the clades. So one clade composed by species from Guiana's region, second clade composed by species from the Amazonian drainage, and the third one by species from Southeast Brazil. This genus has 28 valid species, and some cytogenetic studies suggest that at least 3 new species can be observed. During my PhD, I've been exploring these sex chromosomes from several ways, and I described that at least 2 independent evolution of multiple sex chromosomes occurred in the genus. The first one is composed by this linkage group XY1, Y2, a multiple sex chromosome, that we also can identify the ancestral XY. And in the species from Amazonian drainage, the X1, X2Y with the ancestral XY. And we have been showing that these sex chromosomes are different and they have evolved differently with different linkage groups by chromosomal painting and other techniques. So in my PhD, I also characterized the composition of these sex chromosomes because I was trying to explore which sequences have been accumulated on the sex chromosomes. So by cytogenetics, I've been mapping repetitive DNAs, especially ribosomal DNAs and satellite DNAs. But there are a lot of questions still unanswered. So the species identified by cytogenetics are confirmed by molecular markers. The sex chromosome turnovers are also supported by molecular data. Are there other sequences besides the repetitive ones being accumulated on these sex chromosomes? And can we identify shared sex-linked markers between the 2 independently evolved sex chromosome systems? And to answer these questions, I've collected 18 species of Hartia in a data set comprising 14 valid Hartia species, plus the 3 new ones suggested by cytogenetics, and 1 to be used as an alpha group. And I collected 3 males and 3 females so I can screen for sex-linked markers. I used DART sequencing. DART is a complexity reduction method. So we have the genomic DNA, and we use a combination of restriction enzymes that target especially it's a combination of 2 restriction enzymes. One is a frequent cutter, and the second one is a target low copy sequences. That's why we can explore for sex-linked markers. And I collected 282,000 molecular markers from DART sequencing, 95,000 of them comprising ZIPs. And I tried to recover the molecular phylogeny for the group. As I previously mentioned, this genus has 3 main clades that I also recovered here, clade 1, 2, and 3. Also, you can see in the PCA. However, I could also insert the cytogenetically identified species in these phylogenies. So here, Hartia SP1 and Hartia SP3. However, look for the PCA. There's an outlier here. This outlier is Hartia SP2, one of the 3 species identified by cytogenetics. And this species is more closely related in our analysis to Farlowella, to the outgroup, than to the genus Hartia as a whole. And we believe that this has occurred because Hartia SP2 is the only Hartia, together with Hartia adsaveti, that have 62 chromosomes in their parent type. And this diploid number is exclusive to these 2 species. And Kerobin, in 2021, she suggested that the heart of Savage should be placed in a new genus. And we also believe that heart sp2 might be another representative of these new genus. When I screened for sex-linked markers, I could obtain 419 sex-linked markers in total, most of them suggesting an XY-based sex chromosome system, a male heterogametic sex chromosome system, and I observed this in all species but heart sp2. So it's another piece of information to confirm that heart sp2 is probably not a heart chart. And the most interesting thing is that we previously known 8 species with sex chromosomes in Harcha, and now I could add the information about sex chromosomes in more 8 species, so doubling the number of known species with sex chromosome systems. When I use PCA on the sex-linked SNPs, I could obtain a large group that groups most of the species, but 2 main clusters here and here, formed by species with the X, Y1, Y2. And I also believe that this has occurred because of a chromosomal process. So chromosomal painting data show that these 2 sex chromosome systems, the X, Y1, Y2 in Harcha Carvalhoi and Harcha sp1, they are composed only by the linkage group that forms the X chromosome. So a fission process of the X chromosome has occurred, giving origin to the Y1 and Y2. However, in Harcha intermontana, this X chromosome is translocated with chromosome 9. So it has a fusion, and Y1 corresponds to chromosome 9, and the long arms correspond to the Y2, that is the ancestral X. So when we looked for the shared sex-linked markers, I could find shared sex-linked markers between clade 1 and clade 3. So species from Guiana's region and species from Southeast Brazil. And I believe that this has occurred because the origin of the XY of the male heterogametic sex chromosome system might be much more older of the species diversification. And a turnover even has occurred here in clade 2, because clade 2 has only the specific sex-linked markers that I could only find between species from clade 2. So some take-home message in conclusion of my work is that all Harcha species that we screened by now have an XY sex chromosome system or a male heterogametic sex chromosome system. A turnover occurred in the origin of the 2 independent XY multiple sex chromosome systems. Shared sex-linked markers suggests an ancestral XY in the genus, and the genus may not be monophyletic. So I want to thank all the people and institutions that made this work available. I will also be in the poster session today, and my poster is 139. And if you screen, if you capture this QR code, there are some previous publications